DNA stands for deoxyribonucleic acid and is one of the two types of nucleic acid found in our cells. The name describes what the molecule is. DNA is a nucleic acid, has a backbone made of phosphate and deoxyribose. DNA is beautifully intricate and works in complicated mechanisms to control the cell. Let me tell you a little something about cells. We are all made up of trillions of cells. There are around 2.5 billion cells in one of your hands, but they are tiny, so tiny that we cannot see them. If every cell in your hand was the size of a grain of sand, your hand would be the size of a school bus. Each cell has its own job, just like us humans do. Some cells help us detect light and see. Other cells help us touch. Some cells help us hear. Other cells carry oxygen around. Other cells help us digest food by secreting enzymes. There are over 200 cell types in the body. That is 200 different jobs. But how does each cell know what job to do? Well, how does human know what job it has to do? Someone tells us. Our cells are also told what to do, but not by a person or computer. Our cells are told what to do by a very special molecule called DNA. DNA is pretty much the instructions for life. DNA is a record of instructions telling the cell what its job is going to be. A good analogy for DNA as a whole is a set of blueprints for a cell, or computer code telling a PC what to do. It is written in a special alphabet that is only 4 letters long. Unlike a book or computer screen, DNA isn't flat and boring. It is a beautiful curved ladder. We call the shape a double helix. The letters of the DNA alphabet, called bases, make up the rungs, special sugars, and other atoms that make up the handrail. The rungs are very special. Each one has a name, but they prefer to be called by their initials, A, T, C, and G. They don't like to be by themselves, so always pair up with a friend. But they are very choosy about their friends. A and T are best friends and always hang out together. G and C are best friends and they always hang out together. Another way of looking at it is that A, T, G, and C are like jigsaw pieces. A and T fit together. C and G fit together. You cannot force a jigsaw piece to fit in the wrong place. Think of all the words you can spell. I bet there are loads, but each word is made using the same selection of letters. Yes, sometimes we leave letters out. Sometimes we repeat letters. But we always have the same selection of letters. Depending on how we arrange the letters of the alphabet, we can make new words. The same is true in the 4-letter alphabet of DNA. If you look at the length of DNA, you can read out all these letters in a row. These letters make up words that are always 3 letters long. These are called codons. These words make up sentences that the cell understands. These sentences are called genes. Let's talk about RNA. Nucleosides are molecules that join together to make up the structural units of ribonucleic acid. Each nucleotide consists of a nitrogenous base, a ribosome sugar, and a phosphate. DNA carries the genetic or hereditary information of a cell and contains thousands of genes. Each gene has a part in the building of a protein molecule.
Proteins serve as a building block of the cells and execute many cell functions. Information gleaned from the genes determines the exact protein composition and precise function of each cell. You may be wondering what the difference is between DNA and RNA. RNA is very similar to DNA, but the two differs in three ways. Structural: in the cell, DNA is usually made up of two strands, while RNA is made up of a single strand and has much shorter chain and nucleotides. RNA nucleotides contain ribosome, which DNA contains deoxyribosome, which also makes it more stable than RNA. The base of RNA is not thymine, as it is in DNA. Rather,
let's go back. The base of RNA is not thymine. Keep going.
Thymine, as it is in DNA, but rather unracial, which is an unmethylated form of thymine that can absorb light. The combination of two or more objects that together from something new is called synthesis. RNA is synthesized from a DNA by enzymes called polymerase. This process is known as transcription. After transcription, RNA is generally further modified by other enzymes. There are also a number of RNA polymerases that synthesize new strands of RNA from already formed RNA. For example, a number of RNA viruses, such as polio, use a type of enzyme to copy their genetic material. So what are the three types of RNA and what are the roles in protein synthesis? RNA is best known for its role in protein synthesis or the production of new proteins. RNA carries information from the DNA to structures called ribosomes, where the actual protein synthesis takes place in the cell. The information is then translated into proteins. There are three major RNA species that serve different but equally critical functions in the protein synthesis process. The three types of RNA are messenger RNA, transfer RNA, and ribosomal RNA. First of all, messenger RNA. Messenger RNA carries the information from the DNA through the cytoplasm or jelly-like fluids that fill the cell to ribosomes. The information is read as three letters words, triplets called codons. Each codon corresponds to the amino acids are the building block of proteins. Second of all, transfer RNA. Transfer RNA and ribosomal RNA serve the functions of translation. Transfer RNA consists of specialized molecules that feature an amino acid at one end and anticodon at the other end. The anticodon can form chemical bonds with the messenger RNA's three-letter codons. In this way, the transfer RNA acts as the translator by transferring the specific amino acids to a
polypeptide
or change of amino acid with the ribosomes. There are transfer RNAs for every amino acid required for protein synthesis. And finally, ribosomal RNA. Ribosomal RNA makes up the structure of the ribosome. It is the ribosomal RNA that reads messenger RNA's codes and translates the information into protein. The protein produced depends on the amino acid sequence in the code. Now I'm hungry, and I'm going to get something to eat.
Since this video has dragged on enough already, I'll try to keep the rest of it simple and to the point. Next off, we'll be talking about scientists and their discoveries. Alright, let's start off with Gregor Mendel. He noticed that certain traits in pea plants followed a specific pattern. Friedrich Meister discovered nucleic acid, DNA. Frank Griffith proved that DNA is a molecule of inheritance. Aaron Chargaff noticed that there was a pattern that occurred in the 4 bases, which are adenine, guanine, cysteine, and thymine. Oswald Avery found that DNA is the material of which genes and chromosomes are made of. Maurice Wilkins discovered the structure of DNA. Hershey and Chase helped to convince the world that DNA is genetic material. Roswell and Franklin discovered the DNA double helix. Watson and Crick discovered the structure of DNA. At that point, my camera fell. Frederick Sanger discovered the sequence of insulin by amino acids and discovered the sequencing of DNA. And finally, John Sultan understood and explained the dividing of cells. So, the last topic we'll be covering is the human genome, which is the international collaboration research program whose goal was to complete the understanding of all the genes of human beings. All our genes together are known as the genome, and what this is trying to accomplish is find which line of code produces cancer or heart disease, for example, and maybe try and be able to prevent it.